Today we're going to talk about a specific type of active transport known as endo and exocytosis. So we use these methods in special cases when our cells encounter a chemical like cholesterol that's unable to dissolve in water, or a chemical that is simply too large to transport through the membrane, even with a channel or gate protein. We'll use the process of endocytosis to move it into the cell. What endocytosis does is it actually forms a pocket within the cell membrane, and then it folds and encloses the chemical it wants to transport within that pocket, which then breaks off of the cell membrane and becomes a vesicle. What this actually means in terms of words in endocytosis, endo means inward, and cytosis is referring to cytoplasm or cell. So endocytosis really means within the cell, and that's what it's doing. It's moving something literally into within the cytoplasm. Most famous example of this is white blood cells in amoebas. They are commonly observed to surround either food in the case of amoeba or pathogen invaders in case of the white blood cell with their own bodies. It looks a lot like they're trying to eat them, and actually in the amoeba's case they are. But another cell is way too big to try to transport into our own cells. So what they do instead is they use endocytosis to completely surround the cell they're trying to engulf. They bring it into a vesicle, and then they'll insert toxic lysosomes into the vesicle to kill and break down the cell. In the case of the amoeba, that's how they actually get the raw materials they use as food. So when we say a cell is eating, in air quotes, a large particle, something that we would consider to be non-soluble or solid, we actually call it the process phagocytosis. And phago literally means to eat. We also have a mechanism that is similar to drinking, where cells will do this with pieces of or particles of fluids, and we call that process penocytosis instead. And oftentimes what we do with this endocytosis is the cell will actually have a receptor protein on the surface. Now, unlike the transport proteins, this receptor protein doesn't allow something to move through the cell membrane, but what it does is when something binds on to that receptor, it triggers a signal to the nearby cell membrane, telling it to undergo endocytosis. So this is how the cell ensures that it's targeting the correct chemical. And we call this process receptor mediated endocytosis. And that is exactly because we're using a receptor to trigger the endocytosis and make sure it happens in the same place. So this is very common for chemicals like cholesterol. But viruses will take advantage of this. So what most viruses do is they actually trick those receptor proteins into thinking that the virus is the chemical they want to take in. And the virus is large enough that it can trigger multiple of them at once. So it can get a big enough endocytotic vesicle that it can exist within them. Once it moves into the cell, it can then take advantage of that and attack the cell's DNA, turning it into basically a viral factory. The opposite of endocytosis then is exocytosis. Exocytosis allows us to get rid of waste within the cells. So that waste gets stored within vesicles and vacuoles, and then there will be proteins on the surface of the vacuole that when it's time to eject them from the cell, the protein will send a signal helping the vesicle merge with the cell membrane and push its contents out into the extracellular fluid. And sometimes this isn't waste either. Sometimes this allows cells to secrete important enzymes or hormones that it uses as chemical signals to communicate with other cells. So we can see this back at this slide too. C is the exocytosis process. So we have a vesicle here that is existing on its own, but when the time comes, it's actually going to merge with the surrounding cell membrane, and that allows the particles inside of it to exit the cell. And we say, because this is usually a fluid that's being ejected, that they're being secreted as if they were coming from a gland.